always been that there we go, thank you. Each of these vaccine trials have a lot to tell us about how we go forward and how, we continue with the vaccine, with the HIV vaccine search. And so one of the things, again, that Yvette had said when we reviewed, or at least when we were really hearing and discussing about these, the outcomes of the Mosaico trial in January, I think towards the end of January, was she said, I was starting to really like HIV vaccines, and now we are hearing this. And I think this is a message for a number of us who are really behind HIV vaccine research, for our communities, as one of the hopes amongst many interventions that we have and that we are looking for. We are really hoping to have a HIV vaccine sometime in the future. But then at this moment, let us really get to understand how it is that we should be continuing to advocate for HIV vaccines. What is it that we are learning? And we're hearing today, I think as Jim has mentioned, from the protocol chair of the Mosaico study, which is a large-scale HIV vaccine efficacy trial known as HVTN 706 or HPX 3002. This was stopped again because of non-efficacy after the DSMB, which is the Data and Safety Monitoring Board, had their usual review and also really found that whilst the product is safe, it did not meet the predefined criteria for efficacy. Therefore, it was recommended that it should be stopped and trial participants should be informed. And once we hear from Susan Binder, who is the protocol chair, we are also bringing Richard Jeffries, thank you, from TAG, Treatment Action Group, who is also going to give us a perspective and and a take of communities, and in terms of, as well as what it is that we should be thinking, what are some of the questions that really need to be answered, and many other things that is Jeff is going to tell us. And we're grateful to both of them for really being part of this conversation. But most importantly, we'd like to hear from you all. Any remarks, any feedback, any thoughts, any question that we'd like to, you have about vaccine research. And we have many others who have participated here. Thanks to Julie Franks, who also is part of this conversation as a site investigator of record from the Harlem site. So anybody else from Bridge HIV San Francisco, Project Achieve, Project Wish, some of the sites that have taken part in this study, the Alabama Vaccine Research Clinic, and some in South America, as well as in Europe. Please, if you are here, would really like to hear your perspectives, your experiences when you have had to break these results to communities as well as participants. We also have somebody who also was very key in the conduct of this study as a director of community engagement, Stefan Wallis. So all of you and everybody, please feel free to be part of this engagement as you are doing. But I won't waste any time. I will give it over to you, Susan, and thank you for taking time to be part of this engagement this morning. Welcome to the very first Margarita Breakfast Show.
Great, thank you so much. And I'm so sorry I'm not on camera. I don't know what's gone on, but my camera doesn't seem to be working at the moment. I don't know if it's a bandwidth issue or what. So at any rate, I was going to walk you through some of the Mosaico data. And I was going to give you first an overview of the study, then talk a little bit about how we handled prep in the trial, and then talk about study outcomes. I'm sorry that some of the data are embargoed because we're presenting it tomorrow, but I'll try to give you as much of the information as I can, and I'm happy to answer questions in the after I do the presentation. So this is an overview of the study. The goal of the study was really, and the goal of this whole approach was to develop a prophylactic HIV vaccine that could work against globally relevant strains of HIV. You probably know, shown here on the left, that there are different clades or different subtypes of HIV around the world. And what we really wanted to do was to try to create a global vaccine that would work for people all around the world. And so this vaccine used an AD26, an adenovirus type 26, which is kind of like a cold virus, and had a mosaic insert. So it had information about HIV from a variety of different strains of HIV from around the world, and then a GP140 envelope protein that included both a Mosaic GP140 and a clade C GP140. And the goal was to give both of these together, first starting with the Ad26 Mosaic, then giving the trimeric GP140 envelope in the hopes that it would prevent infection, it would protect people from HIV infection. And some of this grew out of what we call the non-human primate studies. So studies in non-human primates in which they tested a variety of different vaccines along with Ad26. So they started with Ad26 given at weeks 0 and 12 or 0 and 3 months. And then at 6 and 12 months, they followed it up either with Ad26 and the GP140, as you see in this vaccine trial that I'm going to be describing, or a variety of other strategies. And you can see that the vaccine that performed the best was this Ad26 followed by Ad26 with GP140. It led to a 94% risk reduction per contact. And this was a rectal challenge study, and there was 67% full protection against HIV, against SIV infection, a related infection, SHIV infection. So HIV, when they tried to tease out what was it that was protective, this vaccine does not produce what we call neutralizing antibodies that actually provide protection against HIV, but rather binding antibodies that work in concert with T cells in a variety of different ways, including through something called antibody-dependent cellular phagocytosis. But all of this doesn't matter. The goal was that there were correlates of what was providing protection. So we knew in the laboratory what seemed to be providing protection for these monkeys, but we didn't, so even without neutralizing antibodies, just these binding antibodies along with these functional antibodies seemed to be providing protection. So here's the final vaccine regimen for Mosaico. It used this Ad26 Mosaic, and it was called a tetravalent Mosaic because it had 4 different components to it. And that was given at months 0 and 3. And then at months 6 and 12, the Ad26 tetravalent Mosaic was given along with this, what we call bivalent GP140, because it had one component was clade C and one component was the Mosaic. And the goal was to enroll a minimum of 3,800 participants. Half were going to get randomly assigned to get the vaccine. Half were going to get randomly assigned to receive the placebo. It was a double-blind study, so neither the study participants nor the staff at the sites knew who was getting which one. And as I said, the doses were either at 0 and 3 months, the Ad26 Mosaic, along with the Ad26 Mosaic and the bivalent GP140 at months 6 and 12, or the other half of the group got the placebo. These are the sites where the study took place in the US and Puerto Rico, Mexico, Peru, Brazil, Argentina, Spain, Poland, and Italy. And I just want you to focus a little bit on Latin America, because that ended up being the majority of the participants that we enrolled in the study. So we started the study in November 2019, and then along came COVID. And so recruitment was paused from March of 2020 until June of 2020, because a number of the sites had to shut down and couldn't enroll participants. And at the start of the pause, there were only 29 participants enrolled, and then enrollment started up again. And in September of 2021, enrollment was complete. And then the last participant completed the 12-month regimen in October of 2022. But so we were fully enrolled by September of 2021. And that was right around the time that the results came out from the Mosaico trial. So this is the sister trial, and you can see it's got a very similar design. This was, the goal was to enroll 2,600 women, same vaccine regimen, except that instead of giving a GP140 that had clade C and Mosaic, it just had a clade C GP140. So there were some differences between the trial. This is a study in women in Southern Africa, and I'll show you the sites in just a minute, whereas the Mosaico trial was a study in men who have sex with men and transgender individuals. So the Imbokodo study did enroll 2,600, a little over 2,600 women at 23 sites across 5 countries. And the study took place from 2017 to 2021. And unfortunately, what was found was that there was no efficacy from this vaccine. So if the red line here is the vaccine, what we had hoped was each time the curve rises, it means that there's been an infection in that arm of the study, and the blue line is the placebo group. What we would have liked to have seen was the red line just staying flat along the bottom and people being completely protected. But what we saw was that the red line and the blue line were not significantly different from each other, and so there was not efficacy seen in the study. So what do we do at this point? Mosaico is pretty much fully enrolled at that point. There was a data and safety monitoring board for Imbokodo that determined that the study should stop. It was the same data and safety monitoring board for Mosaico, and they said Mosaico should continue. And there were some reasons for that. There were some differences between Imbokodo shown on the left and Mosaico shown on the right. So I pointed out that the Imbokodo trial only used a clade C GP140, whereas Mosaico used a clade C and a mosaic GP140. They took places in different regions of the world, in Southern Africa versus the Americas and Europe. They were different populations. The Imbokodo trial was younger women aged 18 to 35, and Mosaico was men who have sex with men and transgender individuals 18 to 60 years of age. They were protecting against different subtypes. The Imbokodo study was predominantly a clade C, protection against clade C was being tested, whereas Mosaico is testing about a predominantly clade B vaccine. We didn't know, I'm going to tell you in just a moment how we handled PrEP in the study, but because everyone had access to PrEP during the trial, we didn't know what the infection rate was going to be like. And we expected that there might not be a lot of PrEP use in Imbokodo, but we expected it to be somewhat higher in Mosaico. And so we weren't sure what the infection rate was like, but we powered it based on about a 2% infection rate. And this was protecting against intravaginal mainly versus intrarectal mainly. Now, one of the things to note is that that non-human primate model that I showed you earlier was based on intrarectal challenges with a clade B envelope. And so there was some reason to believe that perhaps we could see protection in Mosaico, even though there was no protection seen in Imbokodo. So let's talk about how PrEP was considered. We know that oral PrEP with daily TAF, TDF, FTC, as well as TAF FTC are highly effective against HIV infection. We've known about FTDF, otherwise known as Truvada, since 2010, and we have had guidelines since 2012. We also know that with FTDF that you can use event-driven PrEP or 2-on-1 or on-demand PrEP, where you take 2 tablets 2 to 24 hours before sex, 1 tablet 24 hours after that, and another tablet 24 hours after that, and that's also highly effective in preventing infection. In the open label extension, it was 97% reduction of infection compared to placebo. We also know that long-acting injectable PrEP is highly effective. And that's true both for men who have sex with men and transgender individuals, as well as for cisgender women. So what were we trying to do in the Mosaico trial? Well, what we were trying to do was to find a vaccine that could be effective in a population of people for whom there was not a good prevention option for any of a variety of reasons. And we have a number of good prevention options. We have U equals U, we have condoms, we have PrEP, we have mutual monogamy. And what we were looking for then was to enroll a population of people who didn't yet have a tile in this mosaic, that they didn't yet have an effective, highly effective prevention strategy that would work for them. And we wanted to try to test the vaccine in that population. So because we were enrolling participants at risk of HIV acquisition and PrEP is highly effective, participants currently on PrEP were not eligible to enroll. But we wanted to ensure that this was really an authentic choice being made about participation in the trial. So what we did was we first screened all participants who, all of the participants who were screened for the trial were first counseled about PrEP. And if they were interested, they were linked to low or no cost PrEP services during the pre-screening process. Linkage to PrEP services occurred instead of trial participation, but we first tried to pitch PrEP to all of the participants. After enrollment, participants received HIV risk reduction counseling, including about PrEP, at all of their study visits. And if participants desired PrEP at any time during the trial, they were linked to low or no cost PrEP services or provided PrEP at the trial site, and they remained in the trial. And then all sites prior to study activation had to have written approved PrEP component plans for both the screening and post-enrollment periods. So what happened with PrEP use during the study? We enrolled 3,887 participants. Remember, our target was at least 3,800 participants. A little less than 10% had ever taken PrEP prior to enrolling in the study. People were not on PrEP when they enrolled in the study, but gradually, as we counseled people about PrEP, some people did opt to take PrEP, so that at 24 months into the study, about 10% of the population in the study had decided to take PrEP, and a little less than 9% of the population took PEP at least once, so post-exposure prophylaxis, which was also allowed in the trial. So now I'm going to talk to you first about the efficacy data and then about the safety data. So let's first give you some baseline characteristics of Mosaico. Most of the participants had a male gender identity at screening. 8.5% were 8.5% were transgender or non-binary individuals. The study population was young with a median age of 28 years of age. 87% overall were Hispanic or Latino, and 8% were Black or African American. But in the US, 28% were Hispanic or Latino, and 19% were Black or African American. And then this is the distribution on the right-hand side of the countries that contributed to the study. And you can see that Peru was the high enroller, almost 42% came from Peru, another 22% from Brazil, and approximately 10% from Argentina, and a little less than that from Mexico. So the vast majority of our participants did come from Latin America. So there was this independent data and safety monitoring board that reviewed the data twice a year and in real time as needed to monitor the safety of the vaccine and to determine if there was efficacy, but also to determine if there was non-efficacy or that it wasn't efficacious during the trial. And the vaccine efficacy was evaluated when there was sufficient follow-up time to determine whether or not the vaccine provided efficacy. There were these predetermined stopping rules that the DSMB had that to determine if the vaccine was not effective. And if it met those criteria, then it was recommended that the trial be stopped. And the stopping criteria were that the 95% confidence bound around the efficacy estimate would have to be below 0%, and the upper 95% confidence bound would be below 50%. So that would indicate that the vaccine did not provide at least 50% protection. And the criteria should be met for both the overall study population, what we call the modified intent to treat population, as well as for those who got you got a good chance of having the vaccine work. They got their first 3 doses of the vaccine on time, and they remained HIV negative for at least 1 month after the 3rd dose to give the vaccine time to work. So that's the per protocol analysis. So the per protocol population. So it had to be true for both all of the participants who were in the trial who were not HIV infected at enrollment, and as well as this per protocol group. And on January 12th of 2023, the DSMB met and the stopping rules for non-efficacy were met. They recommended stopping the trial. The governing council, comprised of leadership at Janssen, the HIV Vaccine Trials Network, and the U.S. National Institutes of Health, spoke with the DSMB and also concluded that the trial should be stopped. So the site investigators were informed that the trial was to be stopped, and a press release was issued on January 18th. Participants were then notified whether they got the vaccine or the placebo, and that the vaccine was not effective. Just to be complete, I want to give you a safety summary. There were no safety issues with the vaccine regimen identified. A little less than 80% of the participants in the vaccine group had either local reactogenicity events, which is things like pain in your arm, or systemic reactogenicity events, things like being fatigued or having, feeling kind of punky. Unsolicited adverse events were observed in 37% of the participants in both of the arms of the study. Serious adverse events were seen in about 4% of the vaccine, of both the vaccine and the placebo arms. And we didn't see any cases of what we call thrombosis with concurrent thrombocytopenia or low platelet clotting with low platelet counts. And that was important because this had been observed in very rarely, but had been observed with adenoviral vector COVID-19 vaccines. So in conclusion, the safety profile of the study vaccine was favorable. Reactogenicity was mostly mild or moderate, and it was short-lived. So just to put this in context, what did this tell us about where we were going with HIV vaccines? Well, in 2015, the HIV Vaccine Trials Network set out a strategy to execute a series of efficacy trials to determine whether in the first place, non-neutralizing antibodies like this vaccine produced could be elicited at high enough titer to be useful. And that was true for both the Ad26 GP140 vaccine studies in Mosaico and Mosaico, as well as the Alvac and GP120 vaccine trial, Uhambo or HVTN 702. And on the other hand, we wanted to determine if we could prevent HIV acquisition with broadly neutralizing antibodies, either infused by themselves, that's called passive prophylaxis, or with a vaccine that would induce BNAbs. And that was really being studied through the AMP trials. And what we found was that the non-neutralizing antibody approach was just not successful. And what was successful was the AMP trials, even though overall there was no efficacy, as Tony Fauci described last night, the people who were exposed to virus that was sensitive to this single broadly neutralizing antibody had 75% efficacy. And so it does tell us that broadly neutralizing antibodies can protect, that this non-neutralizing antibody approach is just not successful enough for us to pursue. And so that's where we're heading right now, is through studies of BNAbs and BNAb-inducing vaccines. So this special session that I hope you can attend tomorrow night from 5:40 to 6 p.m., I'll be reporting on results from the Mosaico trial, including PrEP use as measured in dried blood spots, Kaplan-Meier curves showing the infections in the vaccine and the placebo arms, and the infection rates by age and by geographic region. And then that's going to be followed by Larry Corey, who's going to talk about future directions for HIV vaccines. And so with that, I'm going to stop. I just want to thank the Mosaico participants without whom this study would not have been done, and who gave us really important information, because this was an approach that we thought could be promising, and then it gave us a definitive answer that it's not promising enough for us to pursue. Clinical research sites and their communities, the advocacy groups, the study teams, and the study partners. And I'll stop sharing, and thank you very much.
Thank you very much, Susan, and we look forward to the session on Tuesday, at least for those who are able to attend, so that we can continue with this conversation. And Yusuf, we're going to give you an opportunity, but we should just hear quickly from our next speaker, and over to you, Richard Jeffries.
Hi, everybody. Can you hear me okay?
Very well, Richard.
Okay, I haven't made slides, although I'm kind of wishing I did now, so that you had something to look at other than me sitting in my hotel room. Thank you, Susan, for the presentation. You know, I think obviously this is not the news we wanted to hear, and it's tough news. But I think it emphasizes that HIV is a really challenging virus to vaccinate against. You know, this is a virus that's really replicating and exploiting the cells that should be leading the immune response to it. And so as the body attempts to mount a protective response, the virus is able to exploit that to sort of replicate and spread in the body. And that's a really, really tough challenge, as I think we've learned over these decades. And it has additional sort of features that make it difficult for vaccination, including a sort of outer shielded envelope that's very difficult for antibodies to get at. And a lot of variability in terms of mutation and an ability to kind of escape from targeting by the immune system. And I think it's really important that both the public and policymakers understand that there are these differences with pathogens like COVID that make it so difficult to develop an effective vaccine. And this really did look like the best vaccine regimen possible in the absence of broadly neutralizing antibodies. So there was a really strong rationale to learn whether this approach worked. And so the study it can't be considered a failure. It answered a really critical question for everybody that's interested in the important task of making an HIV vaccine. And I think particularly it should be noted that there was a lot of preparatory work and community consultation that was really laudable to ensure that the trial was conducted ethically. It was an incredible achievement in the way that it was able to enroll and answer the question. And the fact that it was stopped is a kind of demonstration that there are always mechanisms built in to ensure that trials don't go longer than they need to if the question has been answered, to ensure that the risk to participants is minimized. Although it's a little bit difficult to sort of think that the seroconversions that occurred might have been avoidable with other interventions. There was a quote from Rafi Landowitz yesterday, that the best PrEP agent he was referring to, but I think this refers to really any biomedical agent, is the one that the person will take and adhere to. And obviously this trial involved people that weren't, the PrEP had they'd chosen a different option. It wasn't what they wanted to be using. And I think there'll still be a lot to be learned from this trial. It's important to understand that because there wasn't protection, we can maybe learn about the correlates of risk of HIV infection and whether people mounted responses to the vaccine that might have been associated with lower risk. But it would be about a sort of their ability of the immune system to mount those responses and not necessarily something created by the vaccine. There's maybe some questions to be answered about the animal models if we have to look at they didn't seem predictive. And there are still some researchers that think that they can be predictive, so that sort of needs to be figured out. And there are some studies still ongoing of non-neutralizing responses, although it sort of doesn't look great for the future PrEP vac in particular. And perhaps there's sort of other possibilities for combining vaccination with other biomedical prevention options. And I think, the sort of unique things that a vaccine can offer in terms of the ease of administration, long-term protection, really make it still important to be trying to develop a vaccine for HIV, and that there are signs of progress in terms of trying to address the difficult challenge of making broadly neutralizing antibodies against HIV, and some other ideas that are still out there, such as a vector based on cytomegalovirus, which has shown some promise. And there's always the possibility that other novel ideas can come along. So I think, it's disappointing. It's been a long slog. I think it's important to have a lot of empathy for all the people, from the study participants to the researchers that have been working at this tough challenge for so long, because it's difficult, but I don't think we can give up.
Oh, Richard, thank you very much. Thank you for those remarks. And I think really the message is that even with the results that we didn't really, that are less favorable, it's still answering a number of questions. And I think you really highlighting that message, which I think is encouraging, and is encouraging all of us to really behind more research to really continue to look for an HIV vaccine. But again, a lot of questions around the infections that we have seen, and what does that mean, and what is it telling us about about the interventions that we are having. Susan is sharing the uptake of PrEP in the study, and that also really raises quite a number of questions, I think, for the field and for all of us as communities and advocates. So we really appreciate those who have raised their hands, and we're just going to go directly to them. But please, also raise your hand. We'd also like to hear from all of those sites who have participated, or community advisory board members who are part of the study, who can share their perspectives. And but for now, we'll go right to Yusuf and Taha to ask their questions. But in the meantime, please raise your hand as well to be part of the discussion. And we're also going to go to the chat to questions and remarks from Khadija. Go ahead, Yusuf.
Okay, thank you so much. Congratulations, Susan. Despite not the good news we wanted, what I feel is actually a nice milestone for science. I'm looking around, it actually gives another perspective to look around. My question is actually just to know, I have been very curious to know what's really the difference or lesson learned from the Mosaico failure, especially coming based on Imbokodo failure. Maybe what was the key difference between those key studies? And I was just worried, basically the Imbokodo was mainly from the Southern Africa, but we can see the Mosaic was really from the American and Europe. What really was brought about changing this cohort? And was there any community activities in the design and also in the trial? Just to know, because yesterday I felt community engagement was one key thing around yesterday response, to make sure this community that actually contribute to this burden, and a lot of things are really carried away. So thank you so much.
Thank you, Yusuf. We'll hand over to Susan.
Yeah, so thank you for the question. Very important. Mosaico and Imbokodo did have those differences that I showed on the chart, but I think that the bottom line that came out of this study with Mosaico is that this approach of non-neutralizing antibodies is just not being successful enough for us to be able to pursue it. That it doesn't seem that we can generate high enough levels of non-neutralizing antibodies that are effective enough to actually prevent infection. So that's the bottom line message that comes out of this study, and that's why we're pivoting to really focus on broadly neutralizing antibodies. There was a lot of community engagement before the trial, during the trial, and there will continue to be after the trial. So we had community is always part of the protocol team. So that was a component of it. I've got some slides, but I wanted to cut back on the number of slides, so I'm not going to go through those in detail. Stefan Wallace is also on the call, and he can speak because he did a lot of community engagement with the study beforehand. We had a community event where we brought together something like 42 members of community, ethicists, clinical trialists, but to talk specifically about this issue of PrEP and how we were going to handle PrEP in the study. We engaged with groups like TAG and like the Black AIDS Institute to get their input and to provide advice on the conduct of the trial. Each of the sites has community advisory boards that they went to. There was community involved in crafting the, as I said, not just the protocol, but the informed consent forms and all of the way that the protocol was developed. And we plan to have a lot of community engagement in disseminating the results and in making decisions about, next steps moving forward.
Thank you very much, Susan. Let's go to Taha. And we have just about 15 minutes remaining before the end of this call. But I think the thing is that we continue this. So please, Taha, just try to be direct with the question, and hopefully we can have also brief responses. Over to you, Taha. so maybe Twaha is still having some connection problems or challenges. Let's go to you, Bill Snow.
Hi, everyone. Sorry not to be there. I wrote my question out in the Q&A, and it's an idea for some way to make lemonade out of this result, and that is that you now have a cohort of people, especially in the Americas, where PrEP is not easily accessible, where you could run an open label study of those same participants and offer them long-acting PrEP. Does anyone have any comments or suggestions about that?
We are actually, Bill, creating a study specifically in Latin America to try to offer different PrEP choices along with PrEP support tools to try to see if we can increase the uptake of PrEP in this population. It's not using the same participants. So that study has ended. It's a great question. We don't have a mechanism for quickly turning around and creating a protocol for to administer to the Mosaico participants themselves, but we are trying to, we know that we need more. Even in the MPrEP trial, which was a PrEP demonstration project, so people were coming in for PrEP in Mexico, Peru, and Brazil, the infection rate in people who didn't take the PrEP at least 4 days a week was quite high. And so it was very high, particularly in younger people and in Peru itself. And so what we know is that we need additional support mechanisms to try to increase both demand for PrEP as well as support for PrEP while people are taking it.
Richard, did you want to respond?
I have to apologize that I was trying to answer a question in the chat, then kind of missed the question.
Well, not a problem. Not a problem. So we'll quickly go to, we just have about 12 minutes remaining before the end of this session today, but we'll try to really bring some of the questions in the chat, and thanks, Richard, for responding to some of those questions. And Julie is telling us that it's really helpful to hear the context about non-neutralizing antibodies. And Patricia, it is very informative. And Khadija's question, I think you have, Richard, to an extent responded to that, but you'd like an explanation on the difference in mechanisms of non-neutralizing antibodies versus PNABs. Well, in short lay person's term, what's behind the difference and or success of one or the other? And I think it just really asking to hear the language so that we're able to translate this to communities. But also, I think is in line with that question, it would be lovely to hear anyone who is here from the sites to really share their experiences of sharing the results. And you have responded to Brian's question about COVID-19 and HIV vaccine and the difficulty. And Mitchell really would like to know what you both think that these results mean for the role of animal studies going forward in HIV vaccine development. So I'll just take those, and any take on any of these remarks and questions.
Sure thing. And so, Brian's question about the difference between SARS and HIV is just really crucial, because I think if you think of a virus like SARS, which infects the lungs, or hepatitis, which infects the liver, the immune system has to come and deal with that virus in the cells that it's infecting, which are different from the immune system. Whereas with HIV, it's infecting and replicating in immune system cells. So the immune system is having to try and deal with something that's inside the immune system itself. And that's a really tough challenge, I think. For the difference between non-neutralizing and neutralizing antibodies, I might defer to Susan on that, if you're on the line, Susan.
Sure. So if we think about neutralizing antibodies are actually neutralizing the virus, it's keeping it from actually replicating and it clears it from the system. Non-neutralizing antibodies bind to the virus, the binding antibodies do, but they act, what we were hoping for was functional antibodies that act in concert with cells. So they don't by themselves clear the virus, but they act by using cells like T cells to clear the virus. And they do that in a variety of different mechanisms that we talked about. ADCP was one of the mechanisms through which basically the T cells can kind of gobble up and eat the virus through the use, through homing with the antibodies. So neutralizing antibodies, though, can work on their own to completely clear the virus out.
And for the animal model question, I think it's difficult and something that really is going to be a topic of ongoing discussion. You know, intuitively, it sort of suggests that that particular model didn't predict what happened. I think there are still, I just saw a paper recently from Veffa Francini's group sort of saying that actually they still think that there is some predictive potential there. So it's something that needs to be figured out. But I think clearly that there was a lack of translation in this case.
Great. We still looking at a number of questions. And I mean, anybody else would still like to raise their hand? Would really like to hear, you know, your voices. We still have some time, and I am really trying to look into questions. But Stefan, would you also like to come in and really give broadly what has really happened in terms of engagement? I think Richard also highlighted a lot of consultations, wide consultations with different stakeholders, because I think this is also a very important aspect. And as Susan has mentioned, before, during, and now after, we continue to engage with communities. Laura, let's please hear from you.
Thanks, Ntando. I should just also clarify that I'm a journalist working for Spotlight in Johannesburg. There's been talk about Mosaico signaling a shift away from a focus on T cells to B cells. And I was wondering if the panelists could talk a little bit about that and what that means in sort of a layman's terms. Thank you.
Sure. So I think there's still interest in T cells. There isn't. So B cells are the cells that actually produce the antibodies. And so there is a focus on neutralizing antibodies and something that we call broadly neutralizing antibodies, because as you know, HIV is very diverse. Even within an individual, there are multiple sort of substrains of HIV. And so what we're looking for are broadly neutralizing antibodies that attack parts of the virus that can't change, so that they're where it's not diverse and where they can it can clear out a majority of the viruses that look like that. T cells are another approach to providing protection against HIV. We think that T cells are still important, and there are vaccines that are currently being developed that are focused more on T cells, like the CMV vector vaccines that Louis Picker has been working on for a number of years. So there is still interest in T cell based vaccines. I think it's this non-neutralizing antibodies that where we haven't seen enough success for us to really pursue that. I did notice there was a question in the chat about, are we throwing out the non-neutralizing antibodies too quickly? Should we be combining that with broadly neutralizing antibodies? And I think that is an important question. But I think right now the goal is, given that we know that broadly neutralizing antibodies can provide protection based on the AMP studies, that we're trying to capitalize on that and see if we can create vaccines that will actually generate broadly neutralizing antibodies, which is a really tough challenge.
Great. Thank you very much. I think I'm going to ask Jim to come and I think Julie, it would really, oh yes, sorry, Laura, Julie, it would be great to hear from you as well in terms of a number of inputs that you're highlighting here. So please come through and I mean, there's a lot of questions that are coming through, but it would really be lovely to hear people's voices as we go towards the end of the session. So I'm going to invite Julie, Jim, Mitchell, as well as Khadija. I know all of the people I called are not very shy.
I missed the question, Tando. Sorry, can you repeat it?
No, I just
heard
needed
from
you. I needed to hear, you know, some of your remarks on the chat box. If we can hear, you know, quickly your points.
I mean, recently I attended a pre-consultation for BNABs, and there were two things that struck me about today's presentation. One, the animal studies, which is why I asked about the predictability of them. And I think it comes down to models. That's what I heard. And some models are more effective than others. I think that's correct. And then the second one is that there's a variety of mechanisms. The simple answer, I suppose, is there's a variety of mechanisms, and we have to, like we're trying to find different combinations of BNABs, we have to find the different combinations of instruments, I suppose. If I were to explain it to a community member, it would be the different kinds of strategies that would help achieve the same goal. So that's just my quick response.
Okay, thank you. And I'm very unfortunate that we really have run out of time, and but really appreciate all of the input that is here, and a lot of remarks and questions about how we go forward, how we reconsider, you know, how vaccine research is done, and all of the different strategies that are out there for consideration. And we apologize we couldn't get to all of those, but I'm just going to give Richard and Susan just last remarks, and then we hand it over back to Jim.
I'll just say thanks to everybody.
Go ahead.
Just thanks
Exactly
to
what I
everybody
was
for joining.
Yeah, thanks for participating early in the morning for some people, and we're hopeful moving on to the next steps. We do need both better uptake of PrEP as well as better vaccines. So stay tuned.
Yeah.
So thank you so much, Susan, Jeffrey, and Ntando, and for all of you for waking up early, coming to this Margarita Breakfast Club. Hope we'll see you in the next 2 days on Tuesday and Wednesday. Tomorrow we're talking about STI prevention. Wednesday will be focused on women and children. And I just want to kind of end with one, maybe a little provocative thing, but I think we all really need to be looking at how we fund HIV prevention writ large. And honestly, vaccines have always gotten the lion's share. And maybe there's a way to think about this more equitably when we think about combination prevention, different strategies tested in the same trials. But I'll just say, as a advocate for HIV prevention, I find myself frustrated that there are certain avenues of research that are not well supported, that are either underfunded or defunded. And so I think we really need to be looking, when we have moments like this of inflection, I think, and as stewards of taxpayer money, much of this is US taxpayer money, we really need to be having hard conversations about how we spend that money, and how we spend that money in the most wise way we can. I know the pie is small, we all don't need to be fighting, but I think if we're all HIV prevention research advocates and writ large, we need to be having tough discussions about how this funding moves forward. So leaving it on a very provocative note, continue having that discussion. I hope to have this discussion with you in person. See a lot of you today. I look forward to seeing people in the community networking zone if you can make it into the poster hall this afternoon during posters. Remember our community WhatsApp for an ongoing chat where that conversation can also continue in real time all the time. And without further ado, folks here in Seattle, there is a plenary in 29 minutes. So wherever you are, take off your robes, take off your slippers, put on something appropriate or not. And we'll see you at the opening plenary and at all the other sessions, hopefully later today. Again, thank you so much. This closes out the first Margarita Breakfast Club of CROI 2023. Ciao.